Okay, I think it's running now. Okay, now, Thursday, remember, is our next little written exercise, which will start with muscle and include nerves and nervous system. The brain is where it stops. We're not going to have cranial nerves. We'll save them for the final. So anyway, the brain is where it stops. 60 multiple choice, 5 short answer. And someone asked this morning about the short answer. Well, some of them are on muscles and some of them are on nerves. So, I mean, you know, variety is the spice of life, as they say. And so we'll chat about the quiz then at the end of class. So, you know, since I've had requests to go over it. All right, today.
I'm sorry, you said the test was through the brain? Yeah,
Through and including the brain. Right, what we're going to talk about now. So, without further ado, there it is. And away we go. All right, now I'm going to kind of skip around here a bit, because some of this stuff we've talked about. All right, when it comes to the size of the brain, this model is actually pretty much life-size. It's about a 3-pound organ. And, you know, it's mostly made of, I know this is such a comforting thought, fat of some sort or other. Now, it's not triglyceride, obviously, but it is made up of different things like phospholipids, ceramides, erythrocytes, gangliosides, a lot of which, and you don't need to worry about this, a lot of which are you know, fatty acids with phosphates or with carbohydrates and so on. So it is largely lipid-based material, is what it is. 4 principal parts, this you never know. Cerebrum, you all know about that. That is actually about 7/8 of the brain. That's the major part, is the cerebrum. And then we have the diencephalon. Well, that's made up of the thalamus and hypothalamus, which is just right in here. It's not a very big part, obviously. And then the cerebellum, which we find back here. And of course, this is all concerned with unconscious activities, particularly that are related to things like balance, posture, equilibrium, etc. And then, of course, when we go on the underside, we have the brain stem, or the medulla oblongata, and the pons, and you know, so there it is. So those are the main parts, all of which are, of course, interdependent, and all of which are quite vital to life as we know it. All right, and then, of course, the brain stem, once it goes to the foramen magnum, becomes the spinal cord, right? All right, when it comes to embryonic development of the brain, it starts very early in life. But it starts originally as what we would call a neural tube. And the neural tube is just kind of like a hollow tube-like structure. All right, and it's fluid-filled. There's fluid in the center of this. So the neural tissue is just really around the outside, kind of like a shell on it. And then it divides into 3 primary brain vesicles. All right, so we have these 3, and they're not all of equal size. There's what can be called a forebrain, and a midbrain, and then a hindbrain, and then the rest of this is still called a neural tube. Now, as you might guess, the neural tube is what will eventually become the spinal cord. All right, so anyway, those are the initial 3 primary vesicles. The forebrain is the top one, that's the one that's the most superior. And again, with these vesicles, they're hollow, so they're filled with the fluid inside. The neural tissue is really just what you find essentially around the edge, around the periphery, like a shell. Now, the forebrain will eventually divide into 2 more sections, and we'll get to them here in a bit. And essentially what that will give rise to is the cerebrum will come out of that forebrain area, and then the lateral ventricles will come from that as well, and then a diencephalon part that will give rise to like the thalamus and hypothalamus, pituitary gland, and so on, and the third ventricle. So all that really comes out of that first vesicle, which is kind of small now, but it does enlarge. The midbrain, which looks like it would be something dramatic, really doesn't produce all that much. It essentially gives rise to what's called a cerebral aqueduct that connects the ventricles. And then the hindbrain, well, that divides into two parts as well. Part of it develops into the cerebellum, part of it then into the pons and the medulla, and then the fourth ventricle comes out of that one as well. And as we said, then the remnant of the neural tube becomes, in essence, the spinal cord. And if we take a look at the fancier diagram, I like my crude ones, right? You know, those are always nice to start with. You know, there was once upon a time I had a student say anytime I wrote something on the board, they never understood it. Well, I don't think they did very well in the class either, as I recall, but anyway. All right. Here we are, it's divided up. There's the forebrain, midbrain, hindbrain, and then the neural tube. And then it continues on. Remember, the forebrain divides into two sections. The top one up here, what's called the telencephalon, don't worry about the term. That will eventually become the cerebrum, as we'll see in the next slide. And then the diencephalon part, which will become the thalamus, hypothalamus, that's that. And then there's the midbrain, and then finally the hindbrain that divides into these two other parts that will give us the cerebellum and the medulla and the pons and all that. So here it is continuing to develop, and you can see now what's happening. It's starting to lap over the top and outgrow the rest of it, and that will become, in essence, the hemispheres of the cerebrum. And so that telencephalon is, in essence, becoming the cerebral hemispheres. Now, when it comes to the coverings, we already know these. I did blindfold you. Tie your hands behind your back, stick your pencil in your mouth, point you to a sheet of paper, and you can write these out. What are the three coverings for the brain and spinal cord? The outermost one is called dura mater, right? And the middle one is called arachnoid, and the final one that covers the brain surface is called the pia mater, right? All right, and with all of these meninges as they're called, the singular form of the word is meninx, but the plural form meninges is what we will use most commonly. And they are, as we've said, the dura mater, the outermost, which actually serves as the periosteum for the cranial bones, as well as the covering of the brain. And it is very dense fibrous connective tissue, a very fibrous layer, as you saw in lab when we were removing it from the brains as we were dissecting them. It has within it, rather, I should say, blood vessels and nerves. All right, and remember that it also actually continues down into like this longitudinal fissure. There's an extension of it that goes in here. We also have an extension of it that in essence comes in toward the cerebellum and then forms also a covering of the cerebellum. Remember having to cut down in there and remove that to peel it out, that falx cerebelli, tentorium cerebelli, and so there we go. The dura mater, as we said, extends down in between the lobes and partitions, and in addition to, of course, covering the brain, it also covers the spinal cord. Now, something that, if you sometimes have a really sharp blow to the head, that you can get is bleeding in the space, and that results in what's called a subdural hematoma. And subdural hematomas turn out to be fatal if they're not relieved because of the pressure it creates on the brain. And in fact, as I mentioned in the other classes, you can even find ancient skulls that have holes drilled in them, you know, to relieve that. So that was a treatment modality that was learned long ago. The arachnoid is that thin, webby-like material, arachnoid, spider-like, literally. And it does not contain blood vessels, and this is the only one of the three layers that doesn't have blood vessels in it. Similarly, it typically doesn't extend down in the divisions either. But the space within it is filled with cerebrospinal fluid. Alright, and then we have the pia mater, which is the thin connective tissue covering right on the surface of the brain. And like the dura mater, the pia mater also contains nerves and blood vessels. And it too, like the dura mater, extends down into the divisions that you find on the brain. Alright, and then we have the spinal fluid. Now, as we said, it circulates through the arachnoid space, but of course it's between both the dura mater and pia mater, and it is a filtrate of blood, and it's a very watery-like fluid, so it's largely water. There's some protein in it, although not a lot, because it is a very watery type fluid, and if it had a high concentration of protein, it would be more like the synovial fluid that you find in joints. It has glucose in it, and it also has urea because it's a filtrate of blood, and that's going to be in any body fluid that is a filtrate of blood, and of course then salts or electrolytes are going to be there as well. And so the whole idea with this is it's there to both protect and nourish to some extent the cells, because some of the cells on the surface actually can get glucose out of that cerebrospinal fluid. The protection business comes from the fact that it acts sort of like a shock absorber, so if there is a blow to the head, that the brain is kind of floating in this material, and it's not quite so likely to be automatically banging up against the cranial bones, causing concussions, although more severe blows can do that, of course. And so there we go. It's found in the ventricles of the brain. Remember, those are the hollow fluid-filled cavities, as well as the arachnoid space, and also down the central canal of the spinal cord in life is filled with cerebrospinal fluid. Now, where is it formed? As we said, in the ventricles, by the filtration of blood, by those capillary tufts called choroid plexes. Now remember, we saw those in lab. We were able to take the probe, remember, and go back in the ventricles and actually pull some of that kind of reddish-brown material out, which was the remnants of the choroid plexes that actually form the spinal fluid. And there we go. Tufts of pia mater are covered by ependymal cells. Remember, the ependymal cells have the tight junctions, and what was the role of those ependymal cells in the ventricles and on the pia mater and other areas in the brain? To form the so-called blood-brain barrier, which was to keep toxic substances, toxic molecules out of the brain, because it's very, the cells and neurons are very susceptible to damage. They're more easily damaged by toxic molecules than are other substances, other cells rather. How much spinal fluid is there? Well, at any given point in time, about around 140, 150 mLs. Actually, there's about 500 mLs produced per day. So it's constantly being produced, and it's constantly being reabsorbed. So where is it produced? It's produced in the ventricles by the choroid plexus. And then it circulates around the brain and the spinal cord. And then where is it reabsorbed? Well, it's reabsorbed by capillaries in the arachnoid called arachnoid granulations. And we'll see those in a diagram here in a bit. So it's constantly being produced, and it's constantly being reabsorbed. Now, occasionally, children may be born that have blockages in the ducts that allow it to circulate in the ventricles. And the ventricles become overfilled and distended, which causes a swelling of the head and actually a swelling of the brain, and that's called hydrocephalus. And hydrocephalus is where there is a blockage that, in essence, causes spinal fluid to remain trapped in the ventricles so that they become distended. Now, needless to say, that's something that does require surgical intervention, because if you put undue pressure on the cerebral surface, on the cerebral cortex, it will cause deterioration on it, which can lead to impaired mental functioning. The treatment is to actually insert shunts into the brain, into the ventricles, to drain off the excess fluid. And usually it's shunted down into the jugular veins is where it goes. And a couple of years ago, I had 2 students one semester that had those. Anyway. So there we go. And we already mentioned this, that the arachnoid granulations actually reabsorb it. Alright, the cerebrum. The surface of the cerebrum has what we call the gray matter, or the cerebral cortex. It's a thin shell, if you will, on the surface of the brain. And of course, most of the cells there are neuroglial cells, but we also find neurons. And the neurons that you find are what we call pyramidal cells. They're large cell bodies, kind of pyramidal shaped, typically multipolar neurons. About 6 cells thick. So it's not a terribly thick area. It's only 4 to 6 millimeters thick. Underlying that are the white matter fibers. And of course, if they're white fibers, what does that mean? Myelinated. And what were the myelinating cells there in the central nervous system? Starts with an O. Oligodendrocytes, that's right. Alright, so there we go. Now, we find that as this grows and as it develops, this outer shell outgrows the white fibers. And that's why it wrinkles up and has this convoluted appearance to it. And what was it we called the convolutions? Gyri. Okay, so a gyrus is a convolution. All the convolutions together would be called the gyri. And because they're outgrowing, and I can't really show you the white fibers here terribly well, because this one doesn't pop off in a cross section, but in any event, there we go. And they're underneath, and they don't grow as fast as the surface, so that's why we get the gyri. But, good thing, because the more of these you got, the more surface area, the more neurons you can pack in there. Now, the deeper grooves are called fissures, and of course, probably one of the most prominent ones, the longitudinal fissure that divides the cerebrum into the 2 left and right cerebral hemispheres. Alright, and then there's another one that divides the cerebellum from the cerebrum. That's also considered a fissure. Then the kind of intermediate grooves, the ones that are not as deep as a fissure but deeper than a gyrus, are called a sulcus in the singular, sulci in the plural. And of course, one of the most prominent ones is here, the central sulcus, that separates the frontal from the parietal lobe. And in fact, we have sulci that separate the various lobes of the brain. Alright. Now, the longitudinal fissure we talked about, and it extends, really, down to about the level of the corpus callosum, which are these white fibers that you see here. The dura mater also extends down into this longitudinal fissure, so it's almost as though the brain is kind of suspended from it. So that's another way of maintaining the brain. And as you also notice here, this cerebral cortex actually extends part of the way down inside this longitudinal fissure. And look at that a little more later when we look at what areas of the cortex control what areas of the body. And between the two, as we said, the extension of the dura mater called the falx cerebri, and that's that extension that goes down in the longitudinal fissure. And you saw that on your brains when you were dissecting them, when you were peeling that off, you could notice the extension that went down into the longitudinal fissure. All right. Now, the frontal and parietal lobes. The central sulcus, remember, divides the two lobes. On the frontal lobe, we have a gyrus that's painted in red on this model. That is called the precentral gyrus. What happens there? That is the location of what we call the primary motor cortex. So, conscious motor impulses that control movements in the body originate here. Alright? So if I want to pick up this model of the brain, well, those impulses are starting there with neurons in that precentral gyrus, which is on the frontal lobe of the brain. Alright, and then in dark blue we have a gyrus on the parietal lobe, because remember the central sulcus separates the frontal and the parietal lobes. That is called the postcentral gyrus, and that is the location of the sensory cortex. Alright, now there are other areas for sensation around the brain, but that is the one that's going to deal with sensing on particularly the surface of the body. And we'll see how that corresponds to different areas. Alright, now the lateral sulcus separates the frontal lobe and the temporal lobe. So here is the temporal lobe in yellow. And then there's a transverse fissure that separates the cerebrum and the cerebellum. That's back here. And the, parieto-occipital sulcus that separates the parietal lobe from the occipital lobe back here. But I don't think those are that vital for us, really. They're kind of minor. I think the central one is by far the one to keep in mind. All right, and then the insula. Now, I can't show you that on this model because in order to get to it, you've got to retract back or open back the temporal lobe. It's buried in here, basically where the frontal, parietal, and temporal lobes all come together. And we'll see it in a diagram here in a bit, so let's just cut the lights a little bit. And here we are looking at the brain. Frontal lobe, parietal lobe, occipital lobe, temporal lobe, cerebellum, transverse fissure would go here to separate the cerebrum from the cerebellum. And then, you know, we have then like that lateral sulcus here, and the parieto-occipital sulcus, and so on. And along here would be the primary motor cortex on the frontal lobe, and the sensory cortex on the parietal lobe. And here again, frontal lobe, central sulcus divides it from the parietal lobe. Precentral gyrus is the primary motor cortex, postcentral gyrus the sensory cortex. And down here the occipital lobe, of course. And here we have the retraction of the temporal lobe to show where that insula goes. It's kind of right there underneath it, really where these three lobes all come together. Frontal, parietal, and occipital is where the insula is located. Alright, a sagittal section of brain. So here we are looking at the cerebrum, and here would be the corpus callosum. Those are white fibers that basically connect the right and left hemispheres. The fornix, the area in between, that's where the lateral ventricle goes. Here's the diencephalon, where the thalamus and hypothalamus are located. And around that would be the third ventricle. And then down here would be the fourth ventricle, which is really between the medulla and the cerebellum. All right, and then that central canal that goes down through the spinal cord. Okay, so I think that gets most. Oh yes, this little extension here, that's an extension of the dura mater, and that's that tentorium cerebelli, and we'll see that again in another slide. Now this is an actual sagittal section of brain. And what gives it that glistening appearance? What layer or covering would give it that? Pia mater, right. And in fact, these blood vessels that you see along here, well, those are actually in that pia mater. Okay? And it's just right on top of the brain tissue. All right, there's the corpus callosum, rather prominent here. There's the fornix, that space between them. That would be one of the lateral ventricles. In fact, this would be the right half, so that would be the right lateral ventricle. Then here would be the diencephalon, so the thalamus is in this area. There, that little open space there, that would be the third ventricle. And remember, as we'll see, it's kind of in the center, under or inferior to those lateral ventricles. And then we have the midbrain, the pons, that enlarged area, the medulla oblongata, and then finally continues on as the spinal cord when it goes through the foramen magnum. Above that, it's still called medulla oblongata. There's the cerebellum, and those white fiber tracts that are in the cerebellum, what are those called? Excellent. The arbor vitae. Yes. Okay? All right, these are the partitions, really, of the dura mater. We talked about these falx cerebelli. Cerebelli. I'll get it out eventually. It's hard to say. That one separates the cerebellar hemispheres, and it's located here. All right, and then we have the falx cerebri that extends down into the longitudinal fissure, and then the tentorium cerebelli that actually come in over the top of, if you will, the cerebellum. Those are the ones you had the fun part peeling off. You said, it's like peeling shrimp. Well, I kind of like that, don't you? Well, I like my food images. All right, and this is just kind of another little summary of what develops out of what. Remember, we had the three primary vesicles of the neural tube. So the three primary vesicles, the forebrain, midbrain, and hindbrain. The forebrain gives rise to the cerebrum, the basal ganglia we'll talk about later, the thalamus, hypothalamus, etc. Those come out of that, along with the lateral and third ventricles. And then the midbrain gives rise to the cerebral aqueduct and the midbrain, which is a very small part. As you notice back up here on the slide, that's essentially all that comes out of that. And then the hindbrain gives us the cerebellum, the pons, the medulla oblongata, and of course the fourth ventricle comes out of that as well. And then the continuation of that neural tube gives rise to then what will become the spinal cord. Another view, and I like this because it shows the ventricles. Here are the lateral ventricles deep within the cerebral hemispheres. There's the third ventricle. And you can see it's right in the center and inferior to the lateral ventricles. And in fact, the thalamus forms the lateral walls of that third ventricle, and the hypothalamus essentially forms the floor and part of the lower walls of it. The gray matter, the cortex, cerebral cortex around the edge, and then the white fibers underneath it. And of course, it's the fact that this outer shell of gray matter grows faster is why and how we get the gyrating, the foldings. Here's the tentorium cerebelli extending over the top, if you will, of the cerebellum. And here we have some of the dura mater extending down into that longitudinal fissure, and in these dural sinuses, that's where the arachnoid villi or granulations will be that reabsorb the spinal fluid. So here's just kind of a close-up view of that. There are the arachnoid granulations. They are capillaries, essentially, that actually reabsorb the spinal fluid. So even though you make about 500 mLs per day, there's only around 140 to 150 mLs at any given point in time. The dura mater and the pia mater, remember, contain blood vessels. The arachnoid does not. But most of the spinal fluid is, of course, in the arachnoid space, in the space in that webby-like arachnoid material. And this is just kind of to show again that the meninges extend down and cover the spinal cord as well as they do the brain. Now the ventricles. As we said, these are cavities within the brain. And we have communication between them. If those means of communication are blocked, what was the condition that can result? Hydrocephalus, yes. All right, so we have then the two lateral ventricles in the hemispheres underneath that corpus callosum, and the third ventricle, which is the slit between the right and left halves of the thalamus. And then those communicate together by what's called an interventricular foramen. So that drains fluid from, in essence, the lateral ventricles and down into the third ventricle. And then the fourth ventricle lies between the brainstem and the cerebellum, and it communicates with the third ventricle by way of what's called the cerebral aqueduct. And of course, a blockage in any of those, foramina or aqueduct would result in hydrocephalus. Now, how does the spinal fluid get out of the ventricles and out into the arachnoid? Well, there are three apertures, if you will, that allow that to occur, and these things are pretty much in the area of the fourth ventricle. So we have the two lateral apertures, and then we have one in the center called a medial aperture, and that allows spinal fluid then to drain from the ventricles out into the arachnoid space to circulate around the brain and spinal cord. Also, some of that fluid will continue on down through the spinal cord and that central canal down to the end of the spinal cord where it will exit into the vertebral canal. And what was that area at the end of the spinal cord called that comes into a cone-shaped structure? We talked about that in lab. Those people in lab. If you haven't had lab yet, you won't know Thursday. Conus medullaris. Remember that? Yes. All right. And another little diagram showing ventricles and the foramina and apertures. Here, looking down on the brain, the lateral ventricles, these are deep within the right and left hemispheres. There's the interventricular foramen, which allows fluid to drain out of the lateral ventricles into the third ventricle. And then the cerebral aqueduct allows it to drain into the fourth ventricle, and then can continue down into the central canal of the spinal cord. And then we have the two lateral apertures and the medial aperture that also allow the spinal fluid then exit the ventricles and enter the arachnoid area to circulate around the outside of the brain and spinal cord. And here's just another view looking at a lateral view. Lateral ventricle, third ventricle, there is the interventricular foramen, and then here's the cerebral aqueduct going down into the fourth ventricle, and then continuing on down into the central canal of the spinal cord. So, there it is. So there's the ventricle system. And all the ventricles basically contain choroid plexes where they can filter the blood and produce the spinal fluid. Most of it's probably going to be produced, of course, in the lateral ventricles because they're the largest. All right. Cerebrum, well, we can divide it up into 3 functional areas, 2 of which we've already talked about, but the third one's very important. There, of course, are the motor areas, the sensory areas, and then to relate the 2 together, we have the association areas. So essentially, association areas are involved with interpretation. Okay? Those are for interpretation. So they really connect sensory and motor. And there we go. Motor areas, as we've said, on the frontal lobes, on that precentral gyrus, all right, and they are large pyramidal-shaped cells, only about 6 cells deep. It's not a very large number of cells when you consider what has to happen there. Impulses travel down the brain stem to the spinal cord on, oh, here's something else to warm the hearts of those that have been in the lab already, corticospinal tracts. Cortico means it starts in the cortex, spinal, it ends in the spinal cord. All right? Now, if it's going from the cortex to the spinal cord, it's descending. All right? All right, if it is a descending tract, well, it's motor. It's a motor type information. It starts in the brain, it goes down to the spinal cord, and it'll go out from there to wherever it needs to go. But in any event, there we are. Now, another thing about most motor tracts, they tend to cross over. So if you're moving your right hand, that impulse actually originated on the left primary motor cortex. You know, and if you're moving your left hand or arm, that started on the right side. It does cross over, and in fact, with those, largely they're going to cross over, in that case, in the medulla is where it happens. All right. There are others like reticulospinal, rubrospinal. Those are unconscious, unconscious. They're motor, but they're there for maintaining posture and balance, and you don't think about those. All right, so there we go. Another little tidbit. Many of these fibers, particularly the ones that are involved in posture and balance, pass through the basal ganglia on their way to the spinal cord. We're going to chat about basal ganglia here in a bit. In fact, we're going to talk about them now. Couldn't remember where I put the slide. All right. These are gray matter deep within the cerebral hemispheres. All right. Now remember, we have the third ventricle in the center. The walls, the lateral walls, the third ventricle, the thalamus. If you move lateral to the thalamus, you find the basal nuclei. That's where they're located. So you've got the third ventricle, then the 2 halves of the thalamus, and then there are 2 of these basal nuclei. One on the right, one on the left. All right? Now, what are the 3 areas of the basal nuclei? The caudate nucleus, the putamen, such a pretty word, and the globus pallidus. Pale globe, literally. All right. Caudate, nucleus, putamen, globus pallidus. Those are the 3 areas that make up the basal nuclei. What is their main claim to fame? They're involved in the production of dopamine. Dopamine is a neurotransmitter, but it is an inhibitory neurotransmitter. What, I wonder, would dopamine do to a section of membrane on a neuron if it were released there? Huh? Well, kind of. It would hyperpolarize that section of membrane. Yes, that's a term we've used before. Now, what do we mean by hyperpolarization of a membrane? It does. It's going to have a higher threshold in order to depolarize. In other words, what it's going to do is it's going to change the resting potential from like a negative 70 millivolts to something lower, like maybe a negative 80 or below. Okay? So it's going to take more of a stimulus in order to get that section of membrane to depolarize. Now, folks who have Parkinson's have problems in this area. They're not making enough dopamine. And therefore, what happens? Well, they get lots of stimulatory impulses. They don't get enough inhibitory ones. And so what happens? They get that jerking motion because they can't exercise fine control. It takes a balance between stimulatory impulses, remember, or what we call excitatory impulse, remember that term, and inhibitory impulses to give us the fine control we need. They're coming out fine with the excitatory impulses. They're just not generating the inhibitory ones. And that's where they get into problems. The cells that produce the dopamine are dying. And they lost a lot of those cells and just not producing adequate levels of it. Alright, so there we go. And here's a diagram that puts all these parts together. So, I don't really like the way we sectioned it, but anyway, we'll get by with it. Alright, here's where the third ventricle would be. So there's the thalamus. There's one half of the thalamus, forming the lateral wall of the third ventricle. Bless you. Here, they're the basal nuclei. Caudate nucleus, putamen, globus pallidus. And if you look at the actual coronal section of brain that we have down in the lab, you can actually see these areas. They do have a different color to them from the surrounding white fibers. Okay? But in any event, that's where they would be. So they're really lateral to the thalamus. And the thalamus is lateral to the third ventricle. So that gives you an idea where they would be. And so a lot of times then, fibers come through this area, down through the basal nuclei area, and that is where, of course, you can get a mix now of stimulatory as well as inhibitory impulses. The Broca's area, which is concerned with speech, it's only on the left, and it's this green area that you see here on the model. So if you have damage to this area of the brain, what would happen? You probably couldn't speak. Okay? Not impaired, you just couldn't do it. Alright, and this is kind of showing, again, some of the things we've talked about on different lobes. Frontal lobe, motor area, motor cortex, central sulcus, postcentral gyrus for sensory areas, Broca's area, etc. We'll fill in some of these other areas here in a bit. Now, this is a very interesting slide. And this is dealing with the motor cortex, which of course is located on the precentral gyrus. Now, what it shows is essentially where certain motor actions originate, and the size of the body part demonstrates essentially how much area is dedicated to controlling it. Now, you notice that it starts down here at the base of the precentral gyrus and then works up all the way to the top and then comes down inside that longitudinal fissure. So, and this is not something to memorize, but here you are there at the very base. There are neurons that control swallowing. Alright, so like the pharyngeal muscles, the tongue, and so on that are needed for swallowing. That's where they're located. It's like the body is inverted. Okay? Then we have things that deal with the mouth, if you will. Chewing, salivation, even speech to some extent, although the main speech center is there, the Broca's area. Facial expression. Here's the hand, much out of proportion to the rest of the arm. Alright? and the thumb even more space than the fingers. Okay, because our opposable thumb is something that helps to make us human, I guess you could say. All right, then we have the forearm, the rest of the arm, the trunk, the pelvis, the thigh, and when you start around the corner, there's the leg, foot, and toes down inside here. Now, one of the things about this, when someone is having surgery in these areas, the individual's kind of left semi-conscious, so they can probe around and see what's being stimulated to know where they are. Because, yeah, you got a pretty good idea that for swallowing it's down here. But now, where specifically is it? It does vary a little bit from one individual to another. And if you're going to be doing any surgery in that area, you better be careful what you cut out and what you separate and so on. So that is necessary. All right, now the sensory areas, they're back here on that post-central gyrus, on the parietal lobe. And they pretty much mirror, as we'll see in the diagram here in a bit, the ones that you found on the motor cortex. Now, when it comes to things like temperature, touch, pressure, pain, well, those are on that post-central gyrus. Now, some other things go elsewhere. Sight, for example, posterior area of the occipital lobe. See the yellow? Back here, that's the visual cortex. That's the visual cortex. That's where sight is registered and actually interpreted in areas adjacent to that. The temporal lobes take care of hearing. Well, that makes sense. That's close to where the nerves come from, for the ears. Taste, well, areas near the base of the central sulcus and along the lateral sulcus. So it's kind of like down in here is where taste is going to register. All right. Smell, well, those are deep down in the cerebrum. Then, like the motor fibers, as we said, the sensory fibers cross. So a sensation that's typically detected on the left of the body registers on the right sensory cortex. And most of the time those fibers cross in the spinal cord. But it, again, depends. I mean, obviously with the eyes, they cross up here in the brain. Remember the optic chiasma? Okay. All right, now, here we are with the sensory cortex. So once again, if you start at the base, tongue and pharynx, and then teeth and gums, and the one I think is most interesting, the lips take up a fair amount of space devoted to them. I mean, that's odd. And then the rest of the face. And then again, when it comes to the hand, the thumb gets its lion's share of area, and the fingers, and the hand a lot more than the arms. Neck, trunk, pelvis, thigh, leg, foot and toes, and look what's down at the very bottom. The genitals. Well, anyway. All right, hidden away in the center. All right, so there you go. Now, association areas. These are the ones that we said, they're not motor, they're not sensory, but they connect the two, and in fact, they're concerned with interpretation. All right, on the frontal lobes, higher intellectual processes. All right, and so things like concentrating, planning, complex problem solving, all that kind of stuff goes there. Now, another area of the frontal lobe we can't miss, and that's what's called the prefrontal areas, right here in front. Now, those are involved with emotional behaviors. And at one time, when you had individuals who had emotional disturbances, they would sometimes be treated with what was called, well, the proper term, frontal-lobotomy. But the more vernacular would have been ice pick surgery. Now, where do they come up with a name like that? Well, they used an instrument that looked a lot like an ice pick. Now, what does an ice pick look like? I don't know if everybody knows what that is or not in the whole world.
A straight, sharp.
Yeah, it's just like a straight piece of metal that has a sharp point on the end, so like you'd have a handle on it. If you're having, a big block of ice here, you'd be chiseling away at it, like that. Well, drill a few holes, and actually they could go through the nose, to be honest. And in you go, and you scramble that part of the brain to destroy it. Well, sometimes that worked, and sometimes it didn't. But anyway, it fell out of use, so we don't see it anymore. But in any event, that was actually a modality that was used, and it was aimed at trying to destroy that prefrontal cortex as a way of correcting their behavior, erratic behavior a lot of times. All right, parietal lobes, those interpret, of course, sensory information, and particularly things about touch, pressure, pain, temperature, those kinds of things. Also aiding in interpreting speech and choosing words and that fit feelings and emotions and things of that sort. The temporal lobes, well, complex sensory experiences like understanding speech and reading are primarily centered there. And also memories of visions, music, and other complex sensations. Quite often that's centered on the temporal lobe as well. The occipital lobes, well, the visual cortex and then the areas adjacent to that for analyzing and interpreting images. Okay, so there we go. And then the general interpretive area, well, that's basically where all the lobes come together. Okay, so those would have neurons involved in essence that would connect between the sensory neurons and the motor neurons. So they're for interpretation is really what they're for. And that's actually a fairly large area that is involved with that. Now everyone has a dominant hemisphere. And in most people it's the left that probably dominates. And that's what is used. And your dominant hemisphere now is used for things like language activities, complex problem solving and thought, that sort of thing. In other folks, the right side dominates, and then there's some where they're equally dominant. Now, that is a little different from being right-brained and left-brained. And here's this little thing, and I'm sure you've all probably heard of this before. In fact, you may have taken some kind of a little test to tell you whether you were right-brained or left-brained. Have you? You haven't? Oh, heavens. It's a shame we don't have more time. We can try it. Well, left-brained people tend to be more logical, sequential, rational, analytical, etc. They like to analyze things, break it into parts, that sort of thing, to understand. Right-brained people tend to be more intuitive and more subjective. They look at the whole, they don't have a need really to break it into parts. They kind of look at the whole and sum it up instantly, sort of thing. They also tend to be more creative. Now, no one is exclusively one or the other. Now, I'm close on conscience, but I still have a little bit on the other side. But anyway, most folks use both of these to some extent, but there's probably one that does dominate. Now, guess what most students are who are very successful at school? Yeah. And guess what most faculty are? They're left-brained. I mean, that's why they can't identify with it. So, but anyway, and so as we note, schools tend to favor the left-brain modes of operation. But but that doesn't mean the right-brain people aren't able to figure things out. They certainly are. In fact, they tend to be more creative. All right. Again, looking at where different things happen on the different lobes. the sensory things and the association areas that go with it. It's just kind of a summary of what we've looked at. Now, memory. All right, now when it comes to memory, we have three kinds that we can define. See, here again, it's breaking things in parts. Short-term, recent, long-term. Now, short-term. Well, where is it centered? It is essentially in the frontal lobe and the thalamus. All right, so part of the frontal lobe and the thalamus involved in that. How long does it last? Well, you know, it could be anywhere from a few minutes to a few hours, perhaps. That's kind of it. Now, if you want to keep it around longer, and to some extent you can consciously determine whether you want to do that or not, it can be moved up to recent memory. Now, recent memory centers around the hippocampus. Hippocampus literally means seahorse, and that's kind of what it looks like. I'll talk more about that one in a bit. All right, anywhere from a few minutes to a few days. That's generally speaking where recent memories, how long they last. Then if you really want to hang on to it, and sometimes even if you don't, it gets moved up into long-term memory. Now the nice thing here is, long-term memory is not stored in just any one place. It's in a number of locations. So it's kind of like with your computer. What do? You back up your drive on the computer, right? So what do? You put things on like memory sticks, you put them on little auxiliary hard drives, portable hard drives. You sometimes put on more than one computer, you know, so if one of them crashes, well, you've still got it somewhere. It isn't totally lost. Well, the brain works the same way. It tends to store long-term memories in a variety of locations, all over the brain. How are the memories stored? It's essentially dendritic connections, just like this. Those little telodendria from another neuron, all right, we can create these little dendritic connections with other neurons, and that's how memories are actually stored. Now, over time, yes, you can lose them, but for the most part, once they're there, they're likely to stay. And I mean, look at people who have dementia. What happens to them? They lose the short-term memory, the long-term, they can still remember things maybe 50, 60 years ago, but what happened, what they had for lunch, they couldn't tell. That's been, you know, long enough interval, it's gone. But yet, ask them what they had for their birthday dinner when they were 13, they probably could tell you. You know, things like that. Because that's in long-term memory. Okay. White matter, well, three types of fibers. Association fibers. All right, associating one gyrus with another, basically. These connect a gyrus on one side of the brain with another gyrus on that same side. All right? So in other words, two gyri connected together by these white fibers that are on the same hemisphere, same side of the brain. Now, next one we have are what are called commissural fibers. All right, like the corpus callosum. That connects the two hemispheres, right and left. Okay? Right and left. So, in other words, something that starts on one side, you know, can also be mirrored on the other side. And then, and as we said, the important groups, the corpus callosum, anterior and posterior commissures, well, that's part of this. So, there we go. All right, and then projection fibers. Well, these allow projection to communicate with different parts of the brain and the spinal cord. So in other words, these tend to be vertical in their orientation. Up and down. Coming up from the spinal cord, going down, you know, into the spinal cord. Those are the projection fibers. Okay, so association between gyri on the same side, commissural between the right and left sides, projection kind of up and down, brain to spinal cord and vice versa. Now, the diencephalon, well, that is, of course, in essence, the thalamus and hypothalamus. They're not very large areas, but they're very important because they tend to be involved with maintaining what we call homeostasis. And what was homeostasis? There's a term we had, I think, the very first day we got together. Maintaining a stable internal environment. Right. All right, thalamus. Two oval gray masses that are found on the lateral walls of the third ventricle. In fact, they form the walls, the lateral walls of that third ventricle. There are two masses that make it up, and then there's an intermediate mass that connects the two together. And you saw that on the sheep brains when we dissected those. Okay? All right, what does it do? Well, it's a relay for sensory impulses except smell. That doesn't go through the thalamus. But if I touch something hot, well, say that's hot, that's going to go to the thalamus. And then the thalamus is going to relay it out to the sensory cortex, so I can tell where it happened. All right, and so it channels impulses in to the proper region of the cortex so they can be interpreted. It just produces a general awareness of pain, touch, temperature, etc. But if you want to localize where it was, there you need the sensory cortex. All right, like if you, reach for a pan on the stove, it's hot, you touch it, all right, and the pain registers, well then you know, say like, well, I did that with my left hand. If you didn't have the sensory cortex function to tell you where it was, you'd know that you touched something hot, but you wouldn't know where. Because the thalamus just gives you kind of a general feeling of these sensations. But to really localize and interpret them, that's where you need the sensory cortex. Now, the hypothalamus is even more involved in many respects. It's not a very large area. The hypothalamus is actually about the size of a grape. Okay? And it's below, essentially, the thalamus, as you get from the name. So, essentially, it forms the floor of those lateral ventricles. Well, no, forgive me. Not the lateral ventricle, the third ventricle. Wash my mouth out with soap. All right. Basically, the floor, essentially the floor of the third ventricle. About the size of a grape, but it does a lot of things that are also quite necessary. Heart rate and blood pressure. That's kind of where the normal value is stored. And so, if there's a sudden drop in blood pressure, that's going to cause an increase in heart rate and constriction of blood vessels. Temperature regulation, that's in the hypothalamus. Water and electrolyte balance, the thirst center. Why do you drink? What makes you thirsty? Any thoughts? Well, if you're becoming dehydrated, what do you suppose is going to happen to the electrolyte concentration of the blood? It's going to go up, all right? And the hypothalamus is monitoring that. And will generate then the sensation, time to drink. Okay? Here you go. And so then you take in water, you start diluting it, and that sensation goes away. Now there's more involved in that, obviously, because if you just took like the flowing garden hose and crammed it down your throat, what would happen to you before there'd be much of a change in the ionic concentration of the plasma? You'd probably burst your stomach, and then you'd croak, and that'd be the end of it. Well, that wouldn't be very nice. So as the stomach stretches, that also inhibits that thirst business, okay? So it's a couple of things happening there. But in any event, that generates kind of the urge,
So your electrolytes go up when you're dehydrated?
The concentration, relatively speaking, goes up because they're dissolved in less water. Okay?
It's backwards to, like on
But it's
marketing.
not, because the thing of it is, if you have a solution, let's say you have a saltwater solution, and you pull out water from it, but you leave all the salt there, what's happening to the salt concentration of the solution?
So why did they market drinks that they're replacing electrolytes for
Isn't that for
dehydration?
sweating?
Yes, because as you perspire, you lose electrolytes.
Okay, so it's not actually tied to the dehydration or your thirst.
Right. Okay. Yeah, because as you perspire, especially if you perspire a lot, you do lose a fair amount of electrolytes.
Okay.
Okay.
Okay, so I guess I was thinking tying the two together and they're being handled separately.
You're thinking then, that's good. But if you just took like a container of, say, of salt and water, and you take out water, which leaves the salt, it's getting more concentrated. And that's what's being registered here, being detected. All right, hunger and body weight, that kind of thing. Digestive secretions and movements. You're sitting there resting and digesting, so that stuff is churning away there in your stomach, and then later it slows down. You don't need the excessive movement secretion. Pituitary gland stimulation, regulation, sleep and wakefulness, circadian rhythm. What was the other gland we had that played a role there? We saw that in lamb. Pineal gland, yes. Okay? All right. And hypothalamus also associated with feelings of rage and aggression. Well, yeah, but not as much as something else. The limbic system is really needed after that. Now, the thing of it is, this is a group of structures, and probably one of the biggies here will be the hippocampus. There are about 5 of them. We'll have a list of them here in a bit. That kind of circle the brainstem. And the thought at one time was, this was sort of a remnant of a primitive reptilian sort of brain. I mean, after all, what do reptiles typically do? Lay around and wait for prey to come by, and then they strike and grab it, right? I think we discussed it in studies. I hate them, but anyway, maybe somebody in there finds them cuddly, I don't know, but anyway. They're not the most intelligent of creatures, right? And they're not exactly nice, they're rather nasty sorts of things. Walk by a rattlesnake, and what's it likely to do? It's likely to strike. It's likely to kiss. I mean, it's fangs out, because it's afraid. But in any event, things that are dealing with emotional aspects that are related to survival, and that's all those things want to do, really. That reproduce, of course. And so we've got feelings of fear, anger, pleasure, pain, and that would mean like sorrowful pain, not physical pain. So in any event, that comes out of the limbic system, and there are a number of structures here. And the hippocampus is one of the big ones. It actually looks a lot like, no, I don't think we do. I'll try and show it to you one of these days. It looks like a seahorse, so that's where the name comes from. The mammary bodies, now we did see those on the brain when we dissected. Remember the infundibulum was, and it was just posterior to that. Little projection there, that was the mammary body. Well, that's part of the limbic system, and so on. Now, the brainstem, well, we have the pons, and that is that enlarged area right here. It's really more pronounced on our brain than it was on the sheep brain. But that's the pons, and the pons is an interesting little thing. It's a lot of white fibers, and it's kind of the switchboard, if you will, of the brain, because it is where fibers that are going to the cerebrum, cerebellum, or going down from the brain to the spinal cord all pass through there. So it kind of routes impulses to the proper area. And so there are the transverse fibers that transmit from the cerebrum to the cerebellum, and then the longitudinal fibers that basically transmit from the cerebrum down to the medulla. So that's primarily going to be motor and sensory information. And then the medulla is just, as we said, continuation of the spinal cord, and it's all white fibers typically. But there are, of course, the gray fibers. There's some gray fibers in there, and cell bodies in there that would also be part of the gray material. And don't worry about the olive, and don't worry about the pyramids. But this is probably something. All right, in the medulla, we find the cardiac center, the respiratory center, vasomotor center. Now, cardiac center, what does it do? Speeds up or slows down the heart rate. Now, respiratory center goes right along with that, because in the respiratory center, if you don't have enough oxygen in the blood, it's going to increase the respiratory rate, but you're probably also going to speed up your heart rate as well. Okay? The two are related. And the vasomotor center goes right along with that because it's controlling blood pressure to some extent by the amount of constriction that it causes in smooth muscle in the walls of blood vessels. So those three things all relate together. Cardiac center, respiratory center, vasomotor center, and they're all part of medulla oblongata, or medulla. Sometimes people just use the term medulla is good enough. All right, and the cerebellum, well, the only thing to say about this, unconscious motor movements for posture, balance, equilibrium. Good enough. The rest of it makes for edifying reading. You can stop there, basically. Okay, I don't think there's anything else up here. All right, so there we go. All right. Now that's more than enough. I think the test is really pretty easy. Now for the quiz. All right, let's take a look at that simple little thing. All right, number one. Oh, wait. Have you turned them in yet?
No.
Well, then why don't you turn them in before we go over it? You remember what you put, right?
Don't work that way.
To be honest, it's common sense. And if you don't say anything horrific, it's correct. Now everybody's all twittered about it, I know, but we'll, oh, okay. All right. No, I was trying to email, I got to sit down and read the things later, but anyway.
Yeah.
Oh,
You didn't have to, no, you didn't have to turn it, you couldn't.
all right.
I just stopped it.
Well, now let's take a look at number one. 3 similarities between skeletal muscle and cardiac muscle. Somebody name one. Striations, right. All right, now, we can go further, but that's good. If there's striations, what gives them a striated appearance? The light and dark bands, right? All right, what is responsible for that? The thick and thin filaments. So they've got that, and they both have the same kind of arrangement, right? Okay, there's one. What else? What's another?
T-tubules?
T-tubules, yes. What else? Sarcoplasmic reticulum, right? What does the sarcoplasmic reticulum do with both? Stores and releases calcium. Now, I think you'll find skeletal muscle actually has a more developed one, but that's okay. But still, they both have. Anything else? Okay, well those will do. That's 3 at least. We have the striations, we have the thick thin filaments, we have the sarcoplasmic reticulum. All right, how do they generate ATP? That would be the same for both. Glycolysis, right, and the Krebs cycle. All right, what about calcium pump? Yes. What about the membrane? Would be very similar, right? What else? Let me think. That'd be a difference. Oh, we've got a number of other things. Oh, myoglobin. Excuse me, they both have myoglobin, right? Okay. All right, now number 2. Now see, that wasn't hard to come up with 3 similarities there, was it?
It's the last one, I think, that we're all stumped on.
Number 3.
Number 3, the third question, I think is the one.
We'll get to it here. All right, number 2. 3 differences between cardiac muscle and smooth muscle. Oh, that's cake. All right, well, for one, striations. They got them in cardiac, smooth muscle doesn't have them. All right, thick and thin filaments. Yeah, they both have them, but they're organized differently. What about the myosin binding heads? You know, the heads on the myosin molecules. In smooth muscle, they're along the entire myosin, right? They're only on the ends in cardiac. Oh, calcium. How do they handle calcium? Cardiac muscle has sarcoplasmic reticulum. What is used in smooth muscle? Well, there's not just sarcoplasmic reticulum. There's also caveoli, and there's also calcium coming in from outside, right? Cardiac muscle tends to generate most of its ATP aerobically, you know, glycolysis and Krebs cycle. What about smooth muscle? Glycolysis only. All right, and remember in cardiac muscle you've got the striated appearance. You don't smooth because the way the myofibrils are arranged. They tend to be more of a cross-hatched diagonal pattern in smooth muscle. They're linear in cardiac muscle. Okay? Now that's easy. Now number 3. 3 differences between contraction of skeletal muscle and a nerve impulse conduction of myelinated multipolar neuron. All right. For one thing, what about the contraction of skeletal muscle? You need calcium release, right? Do you need calcium to get a nerve impulse conducted in a neuron? Not until you get to the synaptic knob, because calcium has to come in to cause the lysis of the synaptic vesicles. But other than that, calcium is not key until you get to that. Okay? But you don't get any contraction without calcium release. Where is the calcium coming from in a skeletal muscle cell? Stored in the sarcoplasmic reticulum, right? Where does it come from in the case of the neuron? From outside. Okay? All right. What about T-tubules? Those are key for skeletal muscle contraction, right? Because what do? They transmit the depolarization down into the sarcoplasm, down into the cell. Do you have anything like that in a neuron? No, you don't. You know, the impulse there spreads strictly along the membrane. And also, you could say, well, there's that sodium flowing through the cytoplasm, but it's just really along the edge. It's not the entire cytoplasm that's being affected. Whereas with muscle, it is. I mean, there's a lot of calcium down in the sarcoplasm. Okay? Another thing, well, all right, what makes contraction occur in the muscle? There are 2 proteins that have to interact. What are they? Myosin and actin, right? Remember that? Thick and thin filaments. Do you have anything like that happening in nerves? No, not really. What's the function of myelin in a nerve? Well, it basically insulates or covers part of the membrane and prevents sodium channels from opening. Do you have anything like that happening in muscle? No, there's nothing comparable there. You know, if you get depolarization in muscle, the entire membrane depolarizes and goes right down into the sarcoplasm, throughout the sarcoplasm. Well, see, there's some differences. Now, that wasn't awful, was it? Well, just, here's the key to this kind of thing. Think of what happens when this, whether you like to be polarizing a nerve or you're contracting a muscle. Think what's happening. And then, is that mirrored in the other type of cell? If it isn't, well, it's a difference. If it is, well, it's a similarity. That's all there is to it. I mean, it's just a little bit of thinking.
Is one involuntary and involuntary?
Well, that is a little hard to say because, yes, you have voluntary control over the muscle, but once you depolarize the membrane, it contracts. You're not going to stop it. Okay? The only control is whether you want it to contract or not, but once it starts, it proceeds. How do you stop depolarization of a neuron membrane? There's a substance that, neurotransmitter that depolarizes, and then there's something that inactivates that neurotransmitter substance. What is it? Well, there's acetylcholine, that's the transmitter, and then what breaks it down or inactivates it? Acetylcholine esterase breaks it down, inactivates it, and then the membrane can repolarize. Okay? But it can't until you get rid of that acetylcholine. Did anybody see 60 Minutes last? Well, it was on Sunday. They did a thing on sarin gas and showed people in Syria that had been gassed with it. Did you see that?
I caught little bits and pieces of it.
Remember what happens? It actually inhibits that acetylcholine esterase. So the muscles contract, they just stay contracting. And one of the keys was if you looked at the pupils, they were greatly constricted. Because they contract, and they stay contracted. Well, anyway, I mean, that was pretty disturbing, but anyway, but well, that fits right in what we talked about. I don't like to see how many people saw that. Well, anyway, the test you'll enjoy immensely, and it is not hard.
You say that every time.
I know, but I know. 60 multiple choice, and maybe it's just home multiple guess. They're not, though. They're easy to figure out. But you got to think a little bit. All right. 60 multiple choice, 5 short answers. You'll enjoy it. See you then, or in lab before. Oh, we just had such a fun lab, though, with the spinal tracts. Didn't you enjoy those?
Yeah. What?
You don't want to
Oh, my
forget your
book.
book.
Yes.
Oh, yes, that would be a good idea.
Thank you.
Uh-huh.
Well, let's see, I suppose we're going to shut off the video.
Oh.
That's okay. And I know you did here, so. You go, shut this off.
Have a good afternoon.
You too.
Thanks.
Well, come on. Well,